Bienvenidos. Bienvenidos.
Welcome, everyone. Welcome. Okay. How are you? How was the weekend? Was it good? Was it a weekend? Did you get outside in the snow? Is it snowing where you are?
In the rain.
Speaking of where you are, we have been learning about where some of you are. I will stop pointing at you because that's really aggressive, isn't it? We have been learning about where some of you are.
In Orangeville.
Orangeville. Mike from Orangeville. We saw you sign in. Hi. That's wonderful. And some of you are not in Canada.
Farther than Orangeville.
Farther than Orangeville.
Like they're in Dunnville.
And some of you are not on this continent. And some of you are not in this hemisphere. Which is delightful, and I'm so glad that you're joining us and that you're able to access this small little 1070 community and all of that stuff. And some of you, I will put my finger away.
Yes, please.
Some of you are in places
Put your fist away.
that are currently experiencing civil unrest and insurrection and violence and horribleness, and I've learned about this over the weekend. Not about the violence, but that some of you are actually in those places. Yeah. So, if your country is currently undergoing a national crisis, please get in touch. Please. And if you want.
It's a good rule of thumb.
Yeah.
If there is martial law, proximity to a civil war, or potentially overthrowing of a government, don't do the squirrel project.
Or just get in touch and maybe not do much more else.
Yeah, because we have ways to, I don't know, relieve that small stress in what must be otherwise a very stressful
all
time.
of this. Yes, get in touch, please.
Now everybo's going online going, what the heck happened in Orangeville?
Nothing. Mike in Orangeville, I hope you're okay.
That council meeting last week, that was a ding-donger.
Hey now, there could be bad stuff going on in Orangeville. Okay. Get in touch, please, and thank you to those who have already. Oh, yay, the flowers. I love the flower. Thank you. These are beautiful. Let's screenshot these and capture them. We've made it through another topic test. One more to go. No big surprises. Don't worry about us changing things on you. I think the only thing that we might add to this last one is a short answer type thing like we had on the first one. But again, it's all going to be the same type of thing. No big surprises. And just taking a preliminary look, y'all did great.
Y'all.
Yeah. Y'all. You all of you folks. So I know I know that students can feel a little unsure, especially when we're talking about applying concepts. It's not that these are particularly difficult. They're not tricky. They're not even particularly difficult, but application is difficult, is challenging. And so it's fine to be not 100% sure, because we only are 100% sure when we've memorized stuff and are able to put it on paper. So, but just looking at it, y'all did great. Okay, when will we have the grades out? You're probably wondering. Well, we still have to finish running the topic test too. So for those who are unfamiliar, there are some students who, for various totally important reasons, still need to do the test, and so they will be doing it today. And then we will shut it down and have it going. We will take a look at every single question to make sure. There are some questions where fewer students got the right answer. That kind of pops up a little red flag for us. And so we take a look at the question to see if it was clearly written and all of that stuff, if there were multiple possible different interpretations and all of that. And so then we make adjustments. And all of those, of course, are going to be in your favor. So give us a couple days. Hopefully by tomorrow, but maybe by Wednesday kind of deal. Okay? Good? Questions? Pop them in the chat. Smith is there. Oh, there we go. Okay, wonderful. Ready?
Did you want to?
I muted you.
Was I not muted?
No, it's just when you click on the waiting room, but someone has already been let in, then it like slides up the list and gives you the button for the first person on the actual join list.
This is how the sausage is made. We don't need to know this. This is just give me the sausage.
Okay.
Sausage, please.
Shall we move on?
No, now I want sausage.
Are there any
eggs and sausage and hash browns at the Old Vienna?
Oh man!
I've now distracted you.
If you are new to Guelph, when the world is less of what it is right now, and going out for breakfast is like a thing you'd like to do, the Vienna restaurant. They make
the Old Vienna.
The perfect poached egg. Yeah.
Like consistently, it's kind of amazing.
Unbelievable. Yeah. Okay. Let's clear. Okay. Ready?
3,
2, 1.
1, 2, 3, 4.
Okay. So, we are going to talk about, this is cool. Now we're going to talk about temperature. So we were talking about conformers and regulators, and remember, that's like a general term that is not variable specific. Now we're going to get into some variable specificity.
Variable specificity, and I love the fact that there are things, as we talk about conformers and regulators, some things can be conformers in one area, regulators in another. Some things can be conformers at low concentrations or low measurements, low densities, low exposures. Yeah. Regulators at higher, like that's, that just speaks to me. That makes me, it makes the hair on my head stand up. It just really excites me about the life. What did Jeff Goldblum say? Life will find a way.
Life will find a way.
Yeah.
Okay, here we go. But first,
I just did, for his benefit, I just did a...
Oh, yeah?
I pounded on the table to make a... ripples in my mug.
Okay. Homework question! Woohoo! And actually, we're going to talk about this homework question, so go. I'll give you a couple minutes. Should we put the music back on?
I could sing.
No. No. Shh, they're working.
Be very, very quiet.
Very, very quiet.
Very pointy.
I was very pointy.
You are, and it's often like right here in front of my face.
In front of your face, that's right.
You're like, and another thing.
Ooh, we're getting close. We're getting close.
Whisper, whisper.
Whisper, whisper.
And that's what the final decision was. Hold on. Oh, wait a second.
What? Wait, what are you doing?
I'm going to write you a note.
You're going to write me a note? Are we passing notes? Check the box. Do you like me?
Shut up.
Okay, just about 30 more seconds. Look what it says. What
Backwards.
should I answer?
Yes.
Yes. A third one.
What? Don't go off script. Shut the fuck up.
Okay, welcome back. Here we go. Enough silliness. No passing notes in class. Is that a thing? Is it texts? Are we old?
Oh, there's no doubt about that. Like,
What does
we
it
are very
say?
definitely old.
Ooh, I just got burned. No.
I have a good lunch.
You have a good lunch.
Yeah.
What do you have?
I'm not gonna. Well,
So, very well done. Super good. We're sharing the results right now. Both the northern and the southern populations are going to experience higher levels of predation for slightly different and delightful reasons, right? So one of them is going to be offset, right, with the background color. Okay, so the southern population is just going to be, there's going to be a greater offset. It's going to be whiter on a blacker background for longer kind of deal. And then the northern population is just going to be white on this darker background for a longer period of time. Okay, so slightly different reasons. So well done. The thing that is awesome about biology is, like, last week, somebody published a paper to show that actually they're not experiencing higher predation in either population
in the Yukon, anyhow.
in the Yukon.
Now, why might that be?
Why might it be? So this is the exact same thing as our fish antifreeze protein kind of walkthrough of the hypothesis and the predictions and the data collection and the flat line of nothingness. So imagine you're a Yukon hair researcher, and you go out and you collect data on predation levels of the northern and southern population, and you get a flat line when your perfectly plausible hypothesis is not supported. What do you do next? What is the next thing that comes to your mind, except for it's all hopeless and I don't belong here and I need to go home, which you shouldn't think. What do you do next?
Oops, time to try again. Yes.
Yes.
And then immediately
There could
something.
be an issue with your methods for
Yep.
sure, right? So yes.
Yeah. Look at the predator population. That's a great idea.
Great idea.
Yeah. And that's what they do. Are they the same levels of predators? Yep. So some ecological hypotheses. Are there any physiological hypotheses out there?
Like what?
What is the predator population eating and when? Yeah. So I think
Yeah.
I agree. I think probably that's what this team. This is like a team of about 20 researchers, some of whom I used to play hockey with, who are now looking at this.
Yeah. Oh, that's a really good point. Science doesn't get done by one individual.
Yeah.
It's like a lot to figure out who's eating hairs. Okay. So yeah, it's like lower the pressure.
Yeah, observational study, get in the field. That's
Yeah.
also great. And in fact, this study was possible because these people, these teams of researchers at different locations across the Yukon have been in the field. So this is actually a time series. It is actually a big one. It's officially, in ecology, basically anything longer than 10 years in a row counts as kind of long term. So this is more than that. It's about 16 years worth of seasonal data.
Yeah.
Could the hares have acclimatized to change their coats faster? Yeah.
Totally.
Yep.
Yep.
And think about, I'm going to plant a seed in a way that,
In a virtual
yeah.
brain.
Remember we talked about, some of you actually had questions about the flounder, about the costs of producing these metabolic strategies. And I think that's where the researchers, anyhow, think about that with regard to the hares. There was a figure last week where we talked about how hares in the Yukon pump out different amount of hair, different lengths of hair, different densities of hair. And so their hypothesis, should I just say it so we can keep going,
Yep.
is that it is just metabolically very costly to do that. And we know that's the case. And what their research, what this time series suggests is that it is so costly that it outweighs whatever potential extra predation might happen. And so there you go. That was unanticipated. And in fact, we're bringing you, this is when we said it was published, it was actually published on a preprint server, so it is not actually out yet. So we're like, I don't know if you can like hold your nose up to the camera,
New
but this
science
is
smell.
yeah, this is the like it's like a new car smell, but it's unpublished science yet.
Yay. Okay. Let's do some learning. Okay, so let's.
You forgot my drinking cup.
Yeah. So here are a couple of terms to throw at you, and again, we're throwing these terms at you to kind of get your brain to like plastify your brain and make it all flexible and go, whoa, okay, wait a second. So there are two strategies when it comes, two like big umbrella strategies when it comes to tolerating the cold. Okay, and you can kind of, well, anyway, let's just go with cold, but heat kind of applies too. So you can avoid freezing or you can freeze. And it's really important to learn these terms, to anchor them relative to the actual biological tissues, okay? Not the temperature, but the tissues, and whether or not they are frozen. If they are not frozen, then you are a freeze avoider. And if they are frozen, then you're a freeze tolerator. So there are things that freeze solid in order to be able to cope with very low temperature. Okay? So we're going to introduce you to these strategies.
And we can see these strategies within, broadly speaking, the same taxa.
Yeah.
So amphibians, there's different amphibians that do one thing, some other species that do the other.
Right? Okay. So a freeze avoider, basically, like this is kind of the strategy that we're most familiar with because that's us, right? We avoid freezing. We put on jackets. We go inside.
We're like Keanu Reeves. We're like, winter comes and we're like, no.
Okay. You know, there are species that migrate. There are species that produce antifreeze proteins. And this is where it's important, the distinction, because the winter flounder is a freeze avoider because it can tolerate freezing temperatures without actually freezing. So it is doing a strategy to avoid freezing, okay? So here's some stuff for you to study to kind of like integrate all of these things. You'll notice different levels of organization, different time scales, all of that stuff in order to kind of get that into your head about, you know, how it all kind of fits together. A freeze tolerator, though, full on freezes, solid, tissue, frozen, like put it in the freezer, take it out, and it will like be alive, okay? And that exists. For some of you, this is probably brand new information. For others of you, you're like, oh my goodness, that's exactly like the blah, and you're right.
And I have some in my freezer.
That's pretty hardcore. Okay.
That's a good pun. Hardcore.
Okay, but first, let's play around with some graphing so that we can understand
But not
some
yet.
things. Not yet? What do you want to do?
Well, just don't start graphing yet.
Don't start graphing yet.
It's going up.
Because we're going to kind of up this a little bit of a level. Usually we give you the axes in order to, and we've just thrown this at you. So like, of course it's going to be like, what are they talking about? But let's try it, okay? So in order to be able to distinguish the difference between a freeze tolerator and a freeze avoider, what could we put on the y-axis?
Put it in the chat.
Put it in the chat. Or like right on the...
No.
What could we put in the y-axis?
The vertical. Some suggestions for tissue temperature.
Okay.
Temperature, body temperature. Body temperature. Temperature they're able to withstand.
Okay. I'm not loving temperature.
Core temperature.
Why am I not loving
Yeah.
temperature? Think about the winter flounder.
Yep.
If we want to be able to distinguish, have two very different lines for a freeze tolerator and a freeze avoider, why is body temperature not the best one?
There's a question.
It's not terrible. It'll give you the difference between regulator and conformer, right?
Yep.
But it won't necessarily get you the difference between a freeze tolerator. I'm all handsy today.
No, it's often, but I just brought it up.
Oh.
Use your left hand.
Okay. It won't necessarily tell you the
Shout
difference
over here.
between a freeze tolerator and a freeze avoider.
There's
So what can we
a
put on
suggestion in the
Yeah.
chat. The temperature should be on the x-axis, perhaps.
Okay, temperature.
that?
Okay, you want to do that?
Movement, metabolism, movement speed, amount, levels of proteins that prevent freezing.
Okay.
Metabolic rate, muscle movement, pulse.
Ooh, pulse. You want to do pulse? Antifreeze proteins, that's good. Yeah, yeah. Heart rate, exactly. Like all of these things, all of these adaptations, right?
Yeah.
Okay, thank you. Yes. So this is what you want to start putting on the y-axis. Let's go directly to the source. And this is like, I'm so gonna like oversimplify now because you've gone like next level. But let's go with number of ice crystals in tissue.
I was just going to say that.
Okay, so here we go. Ice crystals in tissue. Now remember, temperature is going to be colder closer to the origin, right? Over here. And temperature is going to increase over here. So let's do somebody who is a freeze avoider. Number of ice crystals with temperature. Draw me a blue line, please. Or a line, it doesn't matter. If you've already got one. Ice crystals in tissue and temperature for a freeze avoider.
Lovely.
Super good. Yeah. Now, you're really hoping that it's 0. Okay?
Yeah.
So those that are a little bit down, closer to 0, and again, we don't have any numbers on this thing, so whatever. So not only will they not change with temperature, they hopefully won't have any.
So,
okay?
I mean, and this is a great point because sometimes we're looking for the direction without respect to where it intersects with the origin. But at this point, it is actually, it's going to be important.
Okay, now, number of ice crystals with temperature for a freeze tolerator.
Should we clear it?
No, let's just use a different color line. Let's go with like green. Catherine's going red. Green or red, it's fine. It'll be beautiful. There we go. Nice. Good.
Lovely.
Thank you. Well done. Super good.
Now there's a majority and a minority opinion.
What's that?
Well, some went up,
Some went up.
some went across.
Oh, yeah. It's going to go down.
Yeah, of course, but this is important. I think partially that might come about. Just remember with the x-axis, cold is on. Which way am I pointing
Closer
on
to
this
the
screen?
origin.
Closer to the origin. Lower number colder.
Yeah, that's great. Good. Okay. Super simple, right?
Well,
Well, good job.
yeah, say that.
Did you screenshot it?
I did.
Yay!
My finger's in my nose.
Okay, well done. Okay.
That's totally fine. That's why we said it.
You want to tell a story?
Well, this is a great story. This is a story about why.
I'm sitting next to someone that has a PhD in frogginess. So
Yeah.
So you do the talking.
So this is, well, stop. Okay. So this is, as a measure, we mentioned earlier that we are old. So one measure of that is that the taxonomy has changed for this beastie. This is not just a hockey puck, depending on the clarity of your monitor, which you're looking at. Here is a wood frog, which when I was growing up academically, we called Rana sylvatica, which is lovely because wood frog is its common name, and frog of the woods is its Latin name. And so now we've discovered a couple of things taxonomically. Remember, we're always mixing up evolution, ecology, and physiology. So what we discovered about the genus Rana was that it was not monophyletic. And so herpetologists among us have renamed these. And so this is now called Lithobates sylvaticus. It remains the wood frog. It remains the most widely distributed amphibian probably in the world. We can find them south of here. You will find them here. If you own any kind of wet, or your parents, or you grew up on or adjacent to any kind of wet territory, wet treed areas, swampy areas, and in the spring you thought, wow, there's a lot of ducks in there. Lots of ducks that you didn't see land, you didn't see take off, and they all kind of sat in there and went, bop, 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 bop. What you have are wood frogs. Wood frogs are widely distributed because they are one of these amazing freeze-tolerant species with a wide latitudinal and longitudinal distribution. So, because of that, and particularly in the northwestern edge of their distribution, in the NWT in Canada, in Alaska, in the excited states of America, these frogs, it's really important that they do this because they might spend 65% of their life in any year or another in this frozen hockey puck state. And they do that because they are the classic. They are like the archetype of how we understand freeze tolerance. They do it, there's no vital signs, the heart stops. They return when they thaw to normal life within a couple of hours. And they're doing this because they use osmolytes, like there's some listed there, like urea, glycerol, glucose. And they basically, there's a sugary-ish, think of them as sugary rounders. And they take the edges off, the pointy sharp bits that ice or that water will form when it's frozen. And so then they figure out, not thoughtfully, not consciously, but it ends up concentrating and protecting the body from where that ice forms and protecting the cell membranes from how the ice forms. So we've got some stories and some videos about this.
And you can tell that Dr. Smith is getting very excited because our CO2 monitor just jumped up 200 parts per million.
Oh my God! Let me tell you about this. So this is an amazing thing. So the other thing that I wanted to mention, I'll put a link to this. This is a classic study, and Costanzo was not George Costanza, but John Costanzo at the Department of Zoology in Miami University. Which I have to tell you as a side is actually not in Miami. There are people who've arrived at Miami University, which is in northern Ohio. With
going,
like
wow, that
flip
flight took
flops
so
and
little
sandals and shorts, and they're like, "Where's the coast?" And it's like, "Well, it's like I don't know, 1,000 kilometers that way." So they freeze solid. They use glucose as a cryoprotectant, and they dehydrate their organs. And they have an amazing. There's different. It's an amphibian, so they're tadpoles and these eggs. And the eggs have some amazing light and temperature strategies too that I can talk about ad nauseum. But our temperature thing will jump up a level. So what does it look like? I hear you say, right? I hear you say that.
Oh.
It looks like this. Come on, work.
In this amazing video, you can see a frog frozen solid at -3 degrees Celsius. It has no sign of life, no heartbeat, no breathing, no observable brain activity. As the animal gradually warms, life returns. And after he warms, he's right back at it, as if nothing has happened. He's ready to breed and create the next generation of frozen frogs. How do they do it?
How do they do it?
The frog pulls water away from its vital organs and lets it freeze in the in-between spaces. Imagine if we could do this. Suspend our lives and lose all signs of life, then spring back when times are good.
Spring back, get it?
Though we know how it manages to avoid freezing damage, much of this amazing feat remains a mystery,
so there's some
and may
questions
well
in
for
here.
quite some time.
Help me remind me to do that. So isn't that amazing?
Right.
I see some questions popping up. Do they just freeze anywhere? And the answer is kind of yes. They stay in the top layer of the leaf litter. They don't bury themselves. And you probably can throw your answer or your thoughts as to why they would do that in the chat. But probably you know that there's a frost line, right? So the frost will extend X amount of centimeters into the ground, and beneath that it's going to be cool, but it's not going to freeze. And so that's actually there's a vertical line in the soil where amphibians and other insects or other animals, insects and arthropods, those that are freeze tolerant will find a refuge and make a refuge above that line in the freezing zone. And animals that are beneath it will that's what they're searching out, is protection from the frost because they're avoiding it. So that makes me think of splendid hair. Now, this has come up because, as Dr. Jacobs mentioned, I have not just a bald, shiny head, but I have a PhD in amphibian biology. I work now mostly with insects and other arthropods, but back in the late 20th century, I was really excited about amphibians.
Sorry.
It would have hurt my confidence as a young man if I'd met you and you were like, at that point. But anyhow, I have much more confidence now. And so one of the projects that I was working on, this paper that's there, and you can go and look at that. It's now like a historical artifact published 21 years ago this month, or last month, that looked at the induction, so the change, a change in response to environmental conditions of a particular enzyme called Photolyze in the wood frog. And there you can see its scientific name, because I am old, we wrote down as Rana sylvatica. So Photolyze, I'll tell you about just briefly, because why was I doing this? Photolyze, we thought in the early 2000s, the late 1990s, that potentially changes in how much ultraviolet radiation we're striking developing amphibian embryos might be like a silver bullet that would explain why amphibian populations around the world were observed to be declining. Of course, there is no silver bullet. There's a lot of different things. There's fungal infections, there's habitat loss, degradation, destruction, and in some cases, the deeper and more intense penetration of ultraviolet radiation can be one of those, but not in this half of the continent, we'll say. And that's part of what I did as my master's research. Now, so Photolyze repairs the principal form of damage to DNA that ultraviolet radiation causes. Photolyze as an enzymatic system is amazing. Bacteria have it, amphibians have it, we don't. That's one of the reasons we need sunblock. If we had this enzymatic system, we wouldn't need sunblock, because we could just, if we were exposed to the sun, this enzyme harnesses another wavelength of radiation and says, repair that principal form of damage to your DNA. Wouldn't that be amazing? But I digress. In doing this, I needed animal tissue to assay for this enzyme. And to get at animal tissue, generally, as a physiological ecologist, what you have to do is you have to kill your organism. I have some problems with that, and I certainly want to minimize it to make sure that any animals that are dispatched for the science, immortalized in the science that we're doing, are done as humanely as possible and minimizing the number that it happens to. So my brilliant idea was that I would go out in the late nights, on warm, wet nights, and I would sit near where roads intersected pond networks. And I would wait for cars to go by and schmuck amphibians that were crossing the road. And I would collect those schmucked amphibians that life had killed, and then I would use their tissues to do that. This was a good idea, I insist. So what that meant was I would use those frogs found dead on the road, and I would collect them for the tissue. So it meant that I had some macabre kind of young Frankenstein nights where I was going out and collecting those, just like shoveling up, because some of these were very active roadways, shoveling up 6, 7 different species of amphibians into buckets, species-specific buckets, then running home that night and putting them in a freezer for when I was going to complete the assay later on in the lab. So 2 weeks later, I go back and I find my bucket of blue-spotted salamanders and my bucket of spring peepers and my bucket of wood frogs. Some of physiological ecology is kind of macabre. Working at the lab bench, these amphibians that I had watched be hit by cars, there was a friend of mine, another grad student, who came in and said, "What are you doing? This looks pretty macabre, Smith." And I said, "Well, Dave, what I'm doing is blah, 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 what I just told you." And he said, "So why is that one moving?" And I was like, "Ha, ha, ha, that's very funny. He's like, "No, it is." And I looked over, and there was a wood frog. Just as you saw in that video, one of the legs kind of spits out from the frozen puck that it's formed in, and it starts propelling it in a bit of a circle. So this is, consider that situation. This is in the spring. The frog has already overwintered successfully. It has emerged. It has thawed. All the resources to producing glucose and all of these cryoprotectants, essentially they've been used up, we figure. It emerges, it starts feeling randy, and it's going to go to the pond and sing. It was a male frog. And then it's hit by a car. And then I pick it up, put it in a bucket, and freeze it again. Not just at like slightly frozen temperatures, but at minus 20.
I feel like Ryan Reynolds should play the character.
Yeah, exactly. This frog is Deadpool. This frog is definitely Deadpool. I'd like to think that, and there would be a lot of swearing. So, this is a high amount of stress, more than your average amount of stress. So it overwintered, it thawed, it initiated freezing, struck by a car, refrozen, rethawed. So what did I do? I did not pith it. I kept it. I maintained it. He might have been a little bit off, but I then very carefully took him back to a pond where I knew there were female wood frogs, because I thought, if ever there was a frog whose genes needed to move on in this population, this was the one. So I tell you this story partially as a research story, partially as a young scientist seeing things and being surprised and having these WTF, the flounder moments.
With the frog.
What the frog? Oh! This is literally a WTF moment. Because they're out there and you run into them all the time. And I suppose also just pragmatically, if there's ever a news story where there's like frogs near the Peterborough area are now resilient to being hit by cars, it's probably because of this frog.
Okay, wait. For those of you who are part of this, you won't be able to see this right now, but for the recording, I have a request.
Yeah?
I'm wondering, and we're going to put your video
Oh my
right
God.
next to your face in here. You'll see this in the video recording. And can I get you to assume the same posture? There we go. Yeah, there you go.
Can't actually.
Yeah, let's do it. Let's do it. And then look in that direction. No, the other direction, because it's mirrored. There. Yay. Done. Thank you.
Oh man.
That will be in the recording.
When hair is splendid as that, you only get it for a short period of time.
Yeah, there you go. Okay. Unless you're me.
So the pre-adapted for traffic, ha ha ha. But this is also, just as with the flounder and with the polar bear and with the Arctic hare. Remember, in the physiology unit, we're offering up these stories specifically or explicitly in the context of climate change and what's going to happen as isotherms, as these lines of similar temperature at different points in the year march further north. Yeah. So, what is going to happen with a wood frog population, individual, that's stuck in the ground, frozen solid as a puck, not exposed to predation anymore because everything's kind of frozen around that point. But then there's less snow in any particular year. And if there's less snow, there's less insulation in that top, say 20 to 30 centimeters, 15 to 30 centimeters of soil. That frog's going to freeze and rethaw, freeze, thaw, refreeze, thaw, potentially much more. Energetically, that's going to be very expensive. Are they going to be able to do it? That's an open question. You could put in the chat some of your hypotheses as to which direction that should go. Would you see things, changes in the distribution of this species, or changes in the overwintering strategies, or the timing? Remember phenology, the timing of these biological events throughout the year? Like several weeks ago or several lectures ago, when we put up those what's happening across the north in response to climate change, and one of them was changes in phenology. That there were graphs like of historically when species flowered and then contemporarily when they flowered, and they're generally all happening sooner. Is that going to happen to this taxa? Is that going to happen to this species?
So, we have the story of a freeze avoider that avoids freezing physiologically, right? Sort of molecularly. It like makes stuff to prevent itself from freezing. Even though it's in freezing temperatures, it doesn't freeze. So it's a freeze avoider. And we have the story of a freeze tolerator. Okay? What is interesting to note, though, or interesting to ask next is freezing an obligate condition? So obligate means that they have to do it, right? Or else there are some consequences to survival. Or is it facultative? Like, is it kind of optional? And to me, anyway, one of the best ways of kind of giving you an example of the difference between obligate and facultative would be, I'm saying that right? Yeah, okay. Some reason in my head I was like, maybe I'm saying it wrong. Okay, the best way to kind of give you an example would be to look at carnivory between cats and dogs. So dogs are facultative carnivores. It means that they can survive on a diet that has very little to no actual meat. Cats are obligate carnivores. They have to eat meat. And that just has to do with the way that their bodies have adapted in order to break down food. So that's kind of a difference of facultative and obligate. The question is, are wood frogs obligate freezers? And if they are obligate, it means that they have to freeze in order to be able to survive. And so it's not just that they do in order to get through the winter, but that if they don't freeze, something gets out of wonk with their physiology, right? If that's true, and it isn't, but if it's true, then climate change is going to have a different effect than if it's a facultative freezer. Facultative means that it doesn't have to freeze in order to be able to, live a life and reproduce, but that it can if the temperatures get to be that cold. And there are really big differences between those two things. One is definitely more able to adapt. The other one is definitely less able, depending upon the conditions.
Excuse me.
So we have some questions for you to think about. Are there any freeze-tolerant endotherms? Do all endotherms attempt to regulate
Excuse me.
a constant high body temperature? How do endotherms deal with extreme cold? Does it depend on time scale of exposure? Yes, it does. So these are things that you can play around with, that you can graph, that you can learn about, and of course we're going to be addressing them next time. And here is a little bit of a guide to help you kind of work through some of these differences. It's really simple, but it'll hopefully get you started. And again, graph these things or figure out kind of what might work on a different graph to be able to distinguish between them, right? So you want to be able to get two different lines going in different directions. Anything else that you want to add?
I'm just running through some of the questions in the chat. There's a question about insects, and there's actually, we could go on and on. But bees, and so some bees are, I'm going to answer about native bees, like bumblebees. They are tolerant. So
Yay.
I'm just going to put that in there.
Okay. Perfect. Okay. That's it for us. Yeah, that's it for us. So we will bid you farewell. We will open up the student hour Zoom in a few minutes. What I will say, though, is I am expecting a call from our family doctor at some point, but we will be here until that happens, and then we'll just keep it open, and you can keep on talking to each other, and I'll just take my call, and then I'll come back, kind of deal. So we're here for the
We're here
next
for a little while. Yeah,
hour.
I'm actually at I'm at a national conference right now, and a student of mine is speaking later this morning, so I might have to jump out to
Yeah,
that.
but we're here.
We're all doing lots of things, aren't we? So well done. So before
Yay.
we leave, put your right hand up, pat yourself on the back, because
did
this
great.
shit ain't over, and you're all continuing to do well with it.
Take care, everyone.
Smile and wave.